the slides that we were looking last Friday. So just need to share it. One sec. Okay, so this one, yes. So you can see the PowerPoint now. Is it okay? Right. Okay, so did you have a look a little bit to this? We ended up last Friday on this slide. Did you check a little bit what we were saying about the relation? So I make a little summary. It is a modelization of the relation of the possibility, the probability of two proteins to interact each other according to the distance that they are separated when they bound to DNA. So did you remember a little bit? We focused on the right part of the slide. So this little graph, if you see my mouse. So did you remember, or do you have any question on this aspect? I leave you talk because it's quite difficult for me to understand if you are understanding or not this modelization.
I think I understand that graph.
So you understood this graph. So on the X, everybody understood a little bit, at least conceptually. So the X-axis is the distance between the two elements that we are studying. So the cis element, the element on the promoter or on the DNA. And on the Y-axis, it is the probability that the two proteins that can recognize these two elements are able to physically interact each other. And so we saw that there were a maximum of probability of interaction between two proteins when bound to DNA, when they are separated from around 500 base pair. It was because of the flexibility and the bending capacity of DNA. And when the two sequences which are recognized by the protein are further separated, the probability is slowly decreasing. But there was this very important drop in terms of probability of interaction when the distance were getting very small, because it was mechanical constraints on the bending of DNA. So is it okay, at least conceptually? I won't go inside what we started to talk about because it's a little bit difficult, but I will re-explain you a little bit through an example that we are going to see today about what is happening when two sites, so this is most of the time we're going to see that for promoter sequence, when two sites for protein interaction are very close to them, the relation is more complicated because it depends on the side of the DNA. But we will see that through an example. Okay, so just to remind you a little bit. So I go for the previous slide. So this is the model that we saw for a classical RNA Pol2 promoter. And we're going to spend all the afternoon studying DNA protein interaction in order to understand how the activity of the RNA polymerase II can be modulated by specific transcription factors. And we're going to study the DNA binding capacity of some proteins, and we're going to talk about the distances between the data box and these regulatory sequences. So just hold on. So we saw that. Just to finish the few slides on the previous presentation. So I put this slide because nowadays we can use a lot of computer-assisted research in terms of finding specific sequences in the genomes. And here I show you something that is quite interesting if you keep in mind. This is, sorry, okay, I go back to the slide. Wait, I need to move some windows. Okay. So here it's a representation of the sequence similarity, okay, to the human sequence of the same promoter region of the same gene found in different species. In other words, you have one promoter sequence. They don't specify you the name of the gene, but it's not a problem. So the idea in this slide is what can we do with bioinformatics in terms of promoter sequence analysis. So before talking about this slide, if you take the sequence, the coding sequence. So the coding sequence is everything that is after the transcription initiation start till the end of the transcription unit. So the coding sequence, the transcribed sequence contain 5' and 3' untranslated region, the exons and the introns. If you do the sequence similarity from one specific gene from originated from human, and that you compare it with the orthologous sequence coming from other animals, you will see that the sequence similarity or identity are very, very important, because in the coding sequence, you need to keep the same frame for translation, you need to keep the same information in the same portion. What is going to change is the sequence. There will be more diverse if you compare the sequence of the introns, which makes sense because the introns doesn't carry information in terms of protein sequence. But overall, the coding sequence are extremely similar between different species. And in this slide is the same analysis, but the comparison of promoter sequence from the same gene in different species. And what we can see here, so in the y-axis of, there are 4 graphs basically stacked one on the top of each other. So all the y-axis are the percentage of similarity to the human promoter. And on the x-axis there, this is the sequence of the mouse promoter, the chicken promoter, frog promoter, and fish promoter. So if you look, so among all these 4 species, there is only one which is a mammal, which is the mouse. Then after we've got a bird, batracian, and then a fish. So very, very different from mammals. And if you compare the promoter sequence between human and mouse, you see, so it's the more the value on the y-axis is high, the higher the similarity is between the two species, between the two sequences. So you see, when you compare a mouse promoter from a human promoter, you see that there are some regions, if you see my mouse, that are absolutely not conserved, but we found in pink very conserved region between mouse and human. Okay, so it makes sense because it's a kind of sequence similarity which is proportional to the phylogenetic similarity between mouse and humans. But when we look in the same genes, but they are coming from chicken, frog, or fish, what you see is basically you don't have any sequence identity between the human promoter and the promoter from chicken, frog, or fish. It's, if you can calculate, it's something like only 10% of identity, or something like that. But what we found, so we found that this is the pink peaks, that there are some very short sequence. Look at the size, I mean the scale, very short sequence that are extremely well conserved among different type of animals. And so it means that when you are analyzing with the aid of computer promoter sequence coming from different species, you are not looking for a perfect sequence comparison, sequence identity, because what is going to be conserved is only small sequences that are the cis element which are important for being recognized by the transcription factor. So when you, the summary of this slide is when you analyze the similarity of the sequence of the same promoter coming from different species, different organism, the similarity is only located in very short stretch of DNA that are corresponding generally to regulatory sequences. Okay, so this is something that you may be going to use for your research if you are working on promoter structure. It's a very good idea to use bioinformatic tools and all the database that are available nowadays to check for what can be conserved in the promoter you are studying in different species, because it can give you some clues about which region could be important. But nevertheless, take care about this approach, because this is only small sequences that are going to be found, and they are not necessarily linked to the transcription activity. So this is to show you a way to analyze the promoter sequence using databases and computer assisted research. Okay, then what we are going to see is how to study technically the structure of a promoter. So this is a strategy that has always been used to study the idea is we've got a piece of DNA and we know that it's a promoter sequence for your gene of interest, and you want to find which are the sequences that are necessary and sufficient. This is very important. The notion of necessity and sufficient to drive the expression of the gene at the right place and at the right time. So the strategy is always the same. So this is an experimental strategy. It's completely different from the one that we saw just before, which use databases and computer aid analysis. This is an in vivo strategy. So the idea is, I'm going to put the little red star. The idea is once you have a promoter like that, you want to find which part of the promoter is important for driving the expression. So everything, the strategy is quite simple. Is we take the promoter. So promoter, we will consider it as including the transcription start. So it means all the sequence necessary for the binding of the RNA Pol2 complex, plus the sequence which are 5' before, so all the regulatory sequences. The idea is using only this promoter that we can clone in a plasmid that contain a reporter gene. So you all know what is a reporter gene, do you? I decided not to show you a list of reporter gene because they are completely different. The one that we use if we work on yeast, if we work on animal cells, if we work on plant cells or insect cells. Anyway, a reporter gene is a reporter that will allow you to monitor the sites and the inductibility of the promoter that you are studying. So the strategy is relatively simple. You take what we call a full-length promoter. And the biggest difficulty here is to define what we call a full-length promoter. In general, but it's not always the case. In general, most, let's say 95% of the regulatory sequences of a gene are contained within the 1 kb region upstream the transcription stack. So I will show you example with real case studies. So the idea is to take the full-length promoter, clone it in front of a reporter gene, and then ligate that to the vector, and then we transfect this plasmid inside the cell that we want to study. And in order to find out which are the regions that are important to direct transcriptional activity, we make a series of deletion. So it means that from 5' towards the transcription site, we make different versions of the promoter, cutting little by little the promoter region. So this is what is drawing here. So we've got, for example, the construct number 1 will contain the full-length promoter fused to the reporter gene. It is this one at the bottom. Construction number 2 contain the promoter region, but deleted from this part of the promoter sequence, fused to the reporter gene and cloned inside the vector. And then you can check, so and so on. So you cut little by little the promoter till the very beginning of the promoter, so around the tata. Okay? So with this strategy, we have a series of deleted version of the promoter from 5' to 3', so going to the transcription start. And the activity of the reporter gene can be tested in transfection or transient expression in different cells to see if the version of the promoter that is tested, is it active, yes or no, in such the cell type. Is it okay for this strategy? Do you understand? So we cut the promoter and we look at when we lose or we gain activity, so you can identify precisely which are the regions which are important. Okay, so it's a very old strategy, still used nowadays, because there is no other possibility to test in vivo. Okay, this is important because at the end, those plasmids, they are transfected inside cells, and you can biologically test the activity of the promoter. This is the only way. Okay, so we're going to see one example of this type of promoter analysis. So it's coming from work that I did quite a long time ago when I was doing my PhD, and I was studying a promoter. So to summarize the main result in the 3-part figure. So on the top, so I will explain you everything, and I would like that all together we try to reanalyze these results. So first of all, we are working promoter. It's not important for you that you know the name. It's a promoter coming from plants, so it's a gene, a plant gene. And this gene is implicated in the biosynthesis of lignin. Do you know what is lignin in plants? Just one quick question, in order to understand the promoter profile.
The what?
Lignin, L-I-G-N-I-N, lignin.
I personally haven't heard of that ever.
Okay, it's not a problem. So in plants, the plant cells have got a cell wall, a very thick cell wall, and this cell wall is composed by cellulose and lignin, okay, especially in trees. So the wood, for example, in a plant, it's 80% of lignin. So this gene, this gene code is called CCR. I explain you very quickly. Well, anyway, it's you don't need to know the gene. So this gene is called CCR. Okay, it's a gene that is coding for an enzyme that is implicated in the biosynthesis of lignin. Okay, that's all you need to know. And the lignin are only produced in specific cell in plant. So in other words, here we are studying a promoter with a tissue specific expression. Okay, and on the bottom left, here you have the B part of the picture. It's a cross section of a stem of tobacco plant, where you see here the epidermis, here the cortex, Here, it's all the vascular bundle of the plant. And here, it's the middle of the stem. It's called the pith. Okay, whatever. So what you see here, you see that you have blue, some cells that are expressing the reporter gene. So the reporter gene in our case is the GUS gene. Do you all know the GUS gene? Yes or no?
Yes.
So I just shift to the whiteboard to make you a small drawing to explain you what we are talking about. And then we will start the analysis. Okay, it's just to explain you the... Okay, so if you take a plant, sorry. If you take a plant like that, okay, so this is the soil, the roots, and the leaves, okay, basically. The picture that we saw, and we come back after, it's a cross-section of the stem. So the stem is like that. So this is the cross-section. In the middle, you've got what we call the xylem and the phloem. So this is the vascular tissue. So this is vascular tissue. Okay, and the picture in the previous slide is a zoom of this part. So it's for you to understand what we are seeing in the picture. So in this part, in this zoom, what you see is the outside of the stem, so this part, okay? And then we see here in the middle, and I write it in blue, activity of the GUS gene in the vascular tissue of the plant. So this is GUS positive, okay? This is where the promoter, the gene is called CCR, the promoter is on in these cells. Is it okay for everybody now? And we could have this pattern of expression because the construct that has been introduced in the plant, the reporter gene is the GUS gene, and we put in front the promoter of the gene CCR. Okay, and this promoter, so this promoter, as you can see in the picture, is able to drive the expression of the reporter gene, GUS gene, only in the vascular tissue. Okay? Is it clear, or do you want me to explain you a little bit more? It's okay. So this is the typical pattern of expression during development of this gene. So this is the same picture, but just in a very young plant, so it's not important. So now we're going to analyze the table on the top. So the table on the top, there is a lot of data, but it's quite simple to understand. So first, we're going to analyze only the top lines. So the lines that start with delta something, delta, 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 delta. So technically, what has been done? What has been done here, you have delta and then position number. Okay, so this position are the position of the fragment of the promoter that has been fused to the GUS gene. In other words, when you see delta minus 1448 plus 220, it means that the sequence, everything is relative to the +1, the transcription start. So this is our reference in terms of position in the gene. So it means that we took almost 1.5 kb upstream the +1. Then we took a little bit of coding sequence till the position 220, and this fragment was fused to the GUS gene. And some transgenic plant were generated from with this construct, and the table show you where, in which tissue, we can find back GUS activity, so the reporter gene activity. And we have that for different dilation. So if you compare from the first line on the top till the last one here, delta minus 77, you see that all the fragments always end at the same position, plus 220, but the beginning is different because we cut little by little the promoter. So from this experiment, different transgenic plants were obtained with this different construct, and those plants were analyzed and checked for their GUS activity. So it's written with plus or minus signs, and here you've got the name of the tissue where we can find in stems GUS activity or in roots. So I explain you a little bit what does it stands for. So EP stands for epidermis, but you see there is no expression in epidermis, so very, very weak. CO stands for cortex. It is this part of the stem. PHL and XYL stands for phloem or xylem. Phloem and xylems are the two cell types that are present in the vascular tissue. PI means peak, and it's the inside of the stem here. So I propose you that, and for the root, it's exactly the same, but we are not going to analyze the root because it's sufficient to analyze what's happening in the stem. So I'm going to explain you how to analyze the first line of the result that we obtain in stems with this construct. And then I will ask you, how would you analyze the rest of the construct? And then we will talk about this strange construct after. So for the first construct, so basically we take 1.5 kb of promoter sequence fused to the GUS gene. We check for the expression, and you see that the promoter is expressed in phloem and xylems. So in vascular tissue, exactly as we see here in the picture. So this is the pattern of expression of the full-length promoter. What happens if we remove almost the first 500 base pair of the promoter? In other words, instead of starting the construct at minus 1,500, we start at minus 1,000. Do you see any change in the profile of expression?
No.
No.
No. So you see, profile of expression of those two constructs, they are exactly the same, but we remove sequences in the promoter. So the question is, are the sequences that we removed from between these two constructs, are they important for driving the transcriptional activity of this gene?
I think
No.
not.
You remove them, there is no effect. So it's not useful. Is it okay? And if you go on analyzing this, okay, so you see, you remove from 1 kb to minus 900 exactly the same. You remove a big part, so you arrive at minus 324, still the same profile. You remove more and more, and look, this construct, the smallest construct, so minus 119, give the same expression profile as the 1.5 kb construct. Do you see this? Yes or no?
Yes.
Okay, so technically, what does it mean? It means that all the work we did for that, it's completely useless, because it means that all the sequence necessary and sufficient for driving the expression of the gene in the vascular tissue are contained in the promoter 200 base pair upstream transcription start. You see? But look what happens when you compare this construct, so minus 119, and the other one, the last one, which is minus 77. You see that when you delete the sequence contained between minus 190 till minus 77, you lose completely all the expression. Is it clear for you? Okay.
Yes.
So to understand, there is this little drawing which represents you the construct. So we won't talk about the first one. We're going to just to check for the two last one. You see the red spot for my mouse? Yes, it's okay? Okay. So you see on the table that this construct is fully working. So this one is fully working, but the minus 77 doesn't work at all. So the difference between this construct is that we remove this part of the DNA contained in the promoter. So the conclusion there is the sequence that are necessary and sufficient, we don't know yet for the sufficiency, but necessary, are located between minus 119 and minus 77, because when you remove them, you lose completely the activity. Is it clear, this? So it's quite simple, but you will see in the exercise that we will do, which is another case. Actually, it's not an exercise. It's presented as an exercise, but it's coming from publication. You will see that sometimes it can be more difficult, and we will have the example later on with the exercise. So, if this part is clear, now we're going to add a little bit more complexity to the analysis. When we compare the minus 118, which is working, to the minus 77, which is not working, the conclusion is that sequence between minus 190 and 77 are necessary, but we don't know if they are sufficient. Sufficient means autonomous. Okay, so there is another way to another additional experiment that needs to be done to prove for the sufficiency of this sequence in driving the activity. So the idea is I'm shifting to the whiteboard because I need to explain you something. Okay, so I cut this. 1 second. It's not very convenient to do all this online. What? Okay, do you see the sorry, I've got problem for the connection. So from the results, so I will draw you back what's happening. So from the result that we have, so this is the promoter, and we finish at plus 220. And then there is the Gus gene. Okay, so this is Gus. This position is minus 119, and this position is minus 77. And the conclusion was that the region between 119 and 77 contains the elements that are necessary only, not sufficient, to drive the expression in the vascular tissue. So how do you prove that this sequence, which is this sequence only, is sufficient for driving the expression? So what you have to keep in mind is before the +1 here, we have the data box. The data box is the site where the complex, the pre-initiation complex will recognize and bringing the pole 2 there. So from the first experiment, this is only what is important to understand. From the first experiment, all these sequences are coming from the promoter of the gene we are studying. Okay? Is it okay for now? Okay. In order to prove that this sequence here is sufficient to drive the expression as the wild-type promoter, we need to make an artificial promoter. And what can be an artificial promoter, according to you? Do you have an idea? How would you prove that the black box here, the regulatory element, are sufficient to direct the expression at the right place, in the right tissue? How would you do? You have an idea or not?
I don't know.
So listen to me, actually it's quite simple to understand. Maybe it's in order to prove, you understand that when we cut the sequence, we prove that the sequence is necessary. If it works at -119 and if it doesn't work at -77, it means that we remove something that is necessary, but we don't show that it is sufficient. The only way to prove that this sequence is sufficient to drive the expression at the right place is to create an artificial promoter. So the strategy will be the same, will be this one. We take the fragment that we identified as necessary. So -119, -77. Okay, it is this fragment that contains putatively the regulatory sequences. And instead of using the endogenous core promoter, the data box from the CCR gene, we will link to this regulatory sequence an artificial data box and then the reporter gene. So I'm going to write it in green. It means that the piece of DNA which is just after the core promoter, so this is also a data box. And then we put the GUS gene as the reporter at the end. So this is GUS. Do you understand the idea? The idea, it is here what we call a minimal promoter. Minimal promoter means data only. Do you understand? Yes or no? Do you understand what I'm explaining you? Yes.
The difference between the normal CTR promoter and the minimal promoter is that the minimal one only contains the data box, and the original promoter may contain something else.
Exactly. You're right. So I was going to ask you, why do we swap the promoter, the minimal promoter? What you have to keep in mind, so the deletion analysis gives us the information that this sequence is necessary for driving the expression, but we didn't answer if it was sufficient, because we don't know in that context, if around this place, so coming from the promoter of CCR, there are other elements. For example, here, I'm going to put it in gray. Here, maybe we have sequence important for the expression, but we don't know. Because once we cut at minus 77, it doesn't work. So it means that because it doesn't work at minus 77, it is impossible to investigate other regulatory sequences. So from here, we say that minus 119 till minus 77 is necessary. And to prove if it is sufficient, we take this sequence and put it in front of a very minimal promoter that contains only the data box. If this construct works, it means that all the elements that we characterized before, moreover being necessary, they are sufficient. It means that they don't need other sequences. Do you understand the strategy? Okay. So what are the minimal promoter that this type of minimal promoter that we can use? So for plants, we use what we call the minus 55. So it's only Tata Box, 35S promoter. The 35S promoter in plant is 35. It's a promoter coming, it's a constitutive promoter coming from a virus of plant. In mammal cells, we use a minus, I don't remember, I think it's 48, which is coming from SV40. SV40 is a virus, it's a simian virus, 40 gene. So it's also a promoter coming from a virus. So anyway, whatever the model is, I mean, plant, animal, yeast, or whatever, or fish or insect, we have constitutive promoter and minimal promoter. So do you understand this strategy to prove that the sequence on the top of being necessary, it can be sufficient? It's very important to understand that. It's okay? Okay, so I go back and we're going to share the rest of the slide. So, okay, now you see the slide. So we go back because this figure is very dense and there is plenty of information in that. So all the first line of the table, we analyze them. And this is the top of those drawings. The conclusion was the one that we drew before, minus 119 till minus 77 necessary, and you see activity for minus 119, no activity for minus 77. So the last rows of the table and the corresponding drawings. So these are the drawing of the construct that are presented on the bottom of this table. These are the construct to prove if the sequence that we found before was sufficient. Okay, so the strategy, as I explained to you, is once you identify the sequence in the endogenous promoter, you take it and you clone it in front of just a minimal promoter. So the minimal promoter is this little gray box with the +1 there, and it's written minus 55 here, okay, whatever. So what has been done, we're going to look at construct A and B first, with the E as well. So A and B, what is it? It's sequence that we characterized before, minus 119 till minus 77, that has been cloned in both orientations, so endogenous orientation or reverse orientation, in front of the minimal 35S promoter. So these are two artificial promoters that has been constructed with the regulatory sequence in front of a data box only. So we can look the expression profile. So let's start with the control experiment just to show you that this minimal promoter is not working at all because it contains only a data box, okay? So you see construct number E, it's only the minus 5535S, so only Tata box, and you see that in none of the tissue there is expression. So it's the negative control, it's perfect, okay? Then let's have a look to what happens if we introduce the construct A and B, so minus 119 till minus 77 in both orientation, in front of a minimal Tata box. So A and B are those two lines here, and looks what's happening. So no expression at all. So what would have been your conclusion if you had only A, construct B, and obviously the control construct E? What would have been your interpretation? Can you answer this? From the deletion, you managed to conclude that this sequence, minus 111, minus 77, is necessary. With the construction of these two constructs, you want to prove if it is sufficient. You construct them, you analyze them, and you don't have any activity. So what would have been your conclusion on this analysis?
I think that if this region only then is not sufficient for the gene to express.
Exactly. It is necessary, but not sufficient. Do you understand? It's okay? Okay.
Yeah.
Okay, so imagine that at that point, it's not a very good news because you find the regulatory sequence that is necessary, and then when you want to test, it is not sufficient. Okay? And I would like that you analyze, we can take 5 minutes for you if you want, to analyze what's happening on construct C and construct D. So I explain you what are those constructs. One important thing in this drawing is that the drawing is at the right scale. Okay, so it means it's a right scale. So it means that when we identified this region, minus 111, minus 77, okay, we were in that situation. Okay, the regulatory sequence identified, this is the little black box, they are located, okay, with that distance from the transcription start. Okay, when we cut till minus 77, okay, it's 77 base pair upstream the transcription start. But when we, wait, I put the red one, but we fused the minus 119 till minus 77 region to the minus 55, the minimal promoter, the location, the precise location relative to the transcription start of this regulatory element is not localized at the same position than in the endogenous promoter. Can you see it on the drawing? So the endogenous promoter, the regulatory sequence are a little bit more upstream, and the difference, it is 20 base pair, because here we have minus 77 and here we are minus 55. So there is 20 base pair of difference in terms of space. So in order, so maybe what we thought at that time when we had this result was, okay, it doesn't work, but maybe because it's just a question of position relative to the transcription start. Okay, so we generate two other constructs in order to locate artificially the sequence that we identified before at different places relative to the transcription start. And this is what we did for construct C and construct D. So look, construct C and construct D, instead of taking minus 119 till minus 77, we took minus, because it was easier to clone, we took minus 218, so bigger part. We know that there are no sequence important there, but we are going to use this sequence as a spacer till the same position, 77. And we clone this fragment in both orientation. So this is the drawing of the two constructs. So let's compare A, so only 119 till 77, and C, 218 till 77. Actually, what we are doing is reconstructing this promoter and this promoter with the regulatory sequence at the same position. This is what is, if you compare the place, the location of the black box in this construct A and the location of the black box in construct number C, it's exactly the same. This construct A, didn't give any activity. And as expected, construct C doesn't give any activity at all. Okay? Is it okay for now? Okay. But the most interesting result was construct D. So construct D is the same as C, except that the piece of DNA is just clone reversed orientation. And because it's been clone reversed orientation, the relative position of the black box, so black box are regulatory elements, relative to the transcription start in this artificial promoter is exactly the same as the position of the regulatory sequence in the endogenous promoter. So the drawing is at scale, so it means you see endogenous promoter, regulatory sequence are at the same position in construct C. And look what happened in terms of GUS activity, reported gene activity. This construct, so reverse orientation, is working exactly as all the other construct of the wild type promoter. You understand? So when you have this type of answer, you can say, okay, this sequence is necessary for driving the expression of the promoter. And when you have the result like the one obtained in construct D, you can say it is necessary and sufficient. Is it clear for you? Okay.
Yes.
And do you see the importance of the position of the regulatory sequence relative to the data box? Can you see it from this example?
Yes.
And do you see that the orientation has no effect? Because the construct number D, the one which is working, the regulatory element is at the right location, but in the reverse orientation. But it's not a problem for transcriptional activity. Do you understand? So you understand, I hope, that the structure of a promoter is completely different from the structure of a coding region. Because in a coding region, if you inverse the orientation of one piece of coding region, it doesn't make any sense because you cannot translate a sequence which is reversed. But for promoter, the location in the sequence is very important because it's a question of DNA flexibility and the possibility of interacting with the pre-initiation complex containing the POL2. So location is very important, but if you reverse the sequence, so other direction of the regulatory element, it doesn't have any consequence because the DNA, the transcription factor can still bind. It binds it in a reverse way, but it's not a problem because its activity, it can activate the RNA POL2, whatever the orientation is. So when you are working with promoter, you have to change of way of thinking because orientation is not important, position are important, and there is very few nucleotidic identity, as I showed you before with the in silico analysis. Is it okay for this analysis of the promoter? So this is a good example to show you how you identified sequence necessary, sequence sufficient, and from that you can also have all the conclusion in terms of localization is important and orientation is not always important. Is it okay? Yes. Okay, so now we're going I'm going to show you. So we are working. It's the same example. Now we are going to work on. So from this experiment, we identified this region of the promoter for being important. Now we are going to be interested in what are the proteins, so the transcription factor, that are able to bind this domain. So I need to change and we are going to do electromobility shift assay, so EMSA. I will come back on this slide just after. Now, what I'm going to do is to open, another file, the one that I sent you this morning. Okay. One sec, to explain you how we are, what is, sorry, it is this one. Okay. Okay. Can you see the slide? Yes. Okay. So this is one of the 4 slides that I sent you this morning. It's a technical explication of some method, and this one is a kind of summary of this electromobility shift assay. So the idea is it's a very simple and very, very powerful method to analyze interaction between protein and DNA. Okay. So the principle is on the left. Okay, I will explain you quite quickly this. So this is the basic protocol, and then this is some more fine experiment that we can do, and we are going to use this kind of experiment. So the principle is very simple. You have a piece of DNA coming from a promoter. It's exactly the piece of DNA that we just saw before, so the one that is necessary and for driving the promoter expression. So it is labeled with either radioactivity or some cold probes. So that's it. Well, anyway, you label the fragment of interest, and the principle is extremely simple. On one side, you've got the target, so the promoter sequence, and on the other side, you've got protein coming from nucleus, from the cell or the tissue in which the gene is active. Okay, so why do we use nuclear protein? Because all the transcription factors are contained inside the nucleus. So we extract the protein from nucleus, and then we put together the DNA fragment labeled plus the protein. If there is an interaction, you will have a complex that is formed, and we can analyze the formation of the complex by a simple gel electrophoresis. So in that case, there are 2 lines. The first line, so we analyze the autoradiography of those gel because it contains a radioactive version of the fragment. So we always do a lane with only the probe. So this part of the drawing corresponds to the gel precisely, and the bottom part is the autoradiography. So first lane, only the probe. Second lane, we put the probe that has been incubated with the extract, nuclear protein extract. What we do is we always put the probe in excess, not to be limited in terms of recognition. So what we see in the autoradiography, if in the mix of protein, there is a protein that is able to interact with the DNA, the global molecular weight of this complex will be higher than the one of the probe, so the migration will be shorter, and we have a shift in the mobility of the free probe. This is what we call electromobility shift, because the is migrating till the bottom of the gel. And because it is now in interaction with a protein, the molecular weight is bigger, so the migration distance will be shorter. And this is the proof of the formation of a complex made with protein and the probe that you done. So you already saw that, yes or no? It's okay, or?
I understand.
Okay, so from this is the right part of the slide. We can be very, very precise. So I have to explain you for understanding the next slide and the exercise. I need to explain you what is a specific competitor and a non-specific competitor. First of all, we won't look at the last right lane because it's not important. What we call super shift is not important for us for now. So I explain you the 4 lanes from the left to the right. So in every EMS experiment, we do this kind of 4 tests. So the first line, you have on the top of the drawing what's happening in every reaction. So the first lane, as I told you, you only put the labeled piece of DNA. So labeling is at the end. So this is the little red spot there, and this is the piece of DNA of promoter. Okay, when you do the gel migration, the migration distance give you a band after autography, which is there. Second lane, you incubate the probe with the protein extract. As shown here, this is the formation of the complex, so DNA plus protein, and the signal for the unbound probe is weaker because a lot of probe has been bound to the protein. You understand? You follow me? Okay. Now we're going to do 2 competition. So competition, this is very fundamental process in biochemistry in the wide range of biochemistry. Competition can be done with ligand and receptors, can be done with DNA like this. So the idea, and pay attention to one thing. On this line here, the DNA fragment is represented in blue and always carry the labeling, the little red spot, okay? In a specific competition, what we do is we put the probe like this lane, we put the probe labeled, we put the extract, so the orange box is representing the protein, but we add in the reaction in excess the same fragment as the probe, but not radioactive, so without any label. Do you understand? Yes or no? It means in the reaction. So, did you understand this reaction? You put the piece of promoter, you label it radioactively, and you add the protein. If there is interaction between the DNA and protein, you will have a complex that is formed. It's okay for now? Yes? Okay. Now we're going to see what is the specific competition. You redo exactly the same reaction. So the labeled probe, the protein, but you add, and this is what is written here, here, and here, you add in excess the same DNA fragment as the probe, but without the radioactive ends. So you see, this bit of this one and this one, they are represented in this drawing. So it means the same sequence, but only this one is radioactive, the other one are not radioactive. Do you understand? We're going to make a competition of the target, either radioactive versus non-radioactive. If you put the fragment interaction, the fragment non-labeled, so no radioactivity, in excess, the protein that you discovered before, it is not able to distinguish between unlabeled from labeled. So because you put a huge amount of non-radioactive DNA fragment, almost all the protein is going to interact with a non-radioactive fragment instead of interacting with the radioactive fragment. And the consequence of that is that the intensity of the complex observed without any competition will be much, much lower and will disappear. It doesn't mean that the complex is not formed. This is autoradiography. The complex between protein and DNA is formed, like shown here, but it's formed with the version that is not radioactive. So in that case, you don't see it. Do you understand this competition? Everybody
Yes.
understand? Actually, what you are doing in this reaction, you put the protein and you propose the protein, plenty of fragments not radioactive and very few fragments radioactive. So the ratio will do that all the protein, most of all the protein will bind to non-radioactive instead of the radioactive one. Okay? So this is what we call specific competition. After, in order to prove the specificity of the interaction, we need to confirm that with a second competition, which is a non-specific competition. So if you understood the specific competition, you will very easily understand non-specific competition. Basically, we are doing exactly the same, except one thing. The fragment that we add as a competitor, the one that is not labeled, instead of having the same sequence of the probe, we use a different sequence. So you see in this example, the probe is in blue and the non-specific competitor is in green. You can see it? Yes or no? So what is going to happen in this reaction compared to this one? In the first, the specific competition, the protein, because it is specifically able to recognize this DNA sequence, can recognize either the non-labeled or the labeled version of the probe. Okay? But if the protein is only able to recognize the blue DNA, even if you put huge amount of non-radioactive DNA, but with a different sequence from the blue, the protein will always find back the right sequence and will never bind to the other sequence of DNA. And in that situation, the complex is formed back again. You understand the specific competition and non-specific competition. So in other way, what you are doing is you always put the same amount of the probe. The probe is the bit of DNA corresponding to the promoter sequence. First case, specific competition, you add the same sequence, but not radioactive, and you know that the complex disappears. It exists, but it's not radioactive, so you don't see it. Second experiment, and the last one, you do the same, which is a non-specific competition. So instead of putting the same DNA fragment as the probe, the same sequence, you put a different fragment. And if the protein that is able to form a complex with your piece of promoter, it is specifically recognizing this sequence, it will never recognize a sequence of DNA which is different. And so you find back the complex. So remember this, these lines, no protein, size of the probe. You add protein, complex formation. you add the specific competitor, you should have disappearance of the complex, and non-specific competitor, you should have the same complex formation as with no competitor. When you have this type of pattern, it means that the complex that you see is made from a protein and your piece of DNA from a sequence-specific recognition. You understand? Okay. One second. I check for one thing. Okay. So from that, I share back the slides. I don't know, because I work. Excuse me. Okay. Okay, you see back the slides now? Yes, I hope so. Okay, so we go on. Sorry, I just so we go on for this analysis. So okay, so you understood that we are going to study this black region, and this black region, it is so wait, I put the red pointer, and this sequence located between -119 and -77 is the one that is written here. Okay, on the top it's written WT for wild type. So I'm not going to explain you all the competition because you see on this chart, so you've got all the gel shift assay, EMSA that are presented here. On the top, you see in every reaction here, where do we put the protein extract? What kind of probe did we use? So wild type all the time. And what kind of competitor did we use in every single line? We are not going to check for everything because it's quite complicated, and we're going to see that through the exercise that we're going to do in the second part. So just the first line. So when you take this the piece that we identified using the deletion and the artificial promoter. So this fragment, the one that is necessary and sufficient to drive the expression of the promoter, we use that as the probe. So you see the first line is the probe itself. So this is the radioactivity, the radio-labeled piece of DNA, which is here. The second lane, we just add protein extract. So this is extract of protein contained in the nuclei of stems of plant. And what you see that you still have the free probe, and you see that there are two big complexes that appears. Do you see it? So it means that this region, okay, is able to form complexes with protein extract. Then I just want to show you the five next line. Very important. Third line, what do we do here? Is it a specific competition or non-specific competition? Look what we put. We put the probe, obviously, the extract, and we add as a competitor the same fragment as the probe. So what kind of competition it is? The competitor has the same sequence as the probe, so it's a specific competition. It's okay? So, because you put a huge amount of this fragment but not radioactive, the complexes are formed in the gel, but you cannot see them because they are not formed with the radio-labeled fragment, with the probe. So you see, The complex is disappearing when we do specific competition. The fourth lane, this one, where you see the two big complexes, it's exactly the same as the previous one, but the competitor is a fragment of DNA that is completely different from the probe. And you see that the complex, they are formed again. So when you have this type of pattern, you can say, okay, in this probe, there is one complex that is formed, that is remove this complex, disappear when specific competitors are used, and are maintained with non-specific competitors are used. So all in all, it means that this fragment is recognized specifically by a specific transcription factor. Is it okay? Right. So, one question before going on. We'll spend 5 more minutes in this slide, and then we will shift to the exercise, and we will come back on the slide of the lecture later on. So, the 4 first lanes, free probe, addition of nuclear extract, and the formation of the complex, specific competition and non-specific competition. So these 4 lanes, this is the typical profile of the fact that you discover a protein DNA interaction that is sequence specific. In other words, you show that there is, in this region of DNA, a protein that is able to recognize the sequence on that DNA. So a putative transcription factor. So after, I'm not going to analyze all the lane, but I just want to show you the philosophy of the strategy. So the fragment of DNA we use as a probe is the piece of DNA that we identified using the dilation analysis just shown before. But it's quite a big piece of DNA in terms of regulatory element, because it's almost 50 base pair DNA sequence. So the next question that can be asked and answered using this kind of technique is where in this 50 base pair region, where the interaction occurs. In other words, is it on the left of the sequence or on the right of the sequence, because the sequence is quite big. So in order to do that, we can use competition to analyze this sequence more in depth. So I explain you how we did. So on this lane, I put the red one, it's easier. So this is the sequence of the probe, wild type probe. Here, A, B, and C, you've got the sequence of 3 overlapping fragments that are shorter, smaller, but that covers the whole range of the DNA fragment used as a probe. So for example, the fragment A is the 5' part of the probe. The fragment B is the 3' part of the probe, and the fragment C is the middle part of the probe. Okay? Do you follow me for that? So we've got a big piece of DNA used as a probe, and we use 3 overlapping fragments, but we're not going to use them as a probe, we're going to use them as competitors. So do you understand the strategy? So let's take a look. First of all, so the first 4 lane, it is to prove the specific interaction. Then lane number 5, we add in excess the fragment B. So fragment B is the 3' part of the fragment of the probe as a competitor. What is happening? Compare with the wild type competitor, non-specific competitor, and B fragment as competitor. So you see that if you add in excess the fragment B as a competitor, you lose the intensity of the interaction. So it means that part of this sequence, the fragment B, is important in the establishment of the complex formation. Do you understand that? So, sequence B contains part of the sequence responsible for the complex formation. If we look at the next line, the next lane is we use the fragment A as a competitor. Fragment A is the 5' part of the probe. And when you use it as a competitor, you see that the complex disappears compared to without competitor. So it means that this sequence, fragment A, when used as a competitor, you have the disappearance of the complex almost in a similar fashion as the full-length promoter. So what does it mean? It means that this sequence contains some nucleotides that are very important for the complex formation, and so on. Okay? So with this, after you can analyze by yourself, if you the other competition, AM1, AM2, and so on, BM1, BM2, these are mutated punctual mutation on fragment A and on fragment B. So it was, the idea was to identify which nucleotide was important for the complex formation. For example, you take the fragment AM4 as a competitor. AM4 is this one. In this fragment AM4, only the 4 last base at the 3' part are mutated compared to the wild type. And you see that AM4 has the same profile of A. So it means that the mutation that you did in AM4 does not change the function of A. So it means that those nucleotides are not important for the establishment of the complexes. Another example, which is much more obvious, is AM3. You take A fragment as a competitor. So I remember when you use A fragment as a competitor, the complex disappears. But if you take AM3 as a competitor, which is A, but these 4 nucleotides that was G in the wild type, they have been mutated in T. And look what's happened in AM3. You have the complex back again, which means that the fragment AM3 is not able anymore to behave as a competitor like the A fragment wild type. So it means that the nucleotide we mutated there, which are in bold here, are very, very important for the complex formation. If you have any mutation in those nucleotides, like there, you lose completely the formation of the complex. Actually, here you see it because it's as a competitor, but they are compulsory for the establishment of the complexes. Do you understand the strategy like that? Is it okay? Yes or no?
I just need a minute to think.
Yes. The idea is, okay, you find a fragment that is a very good competitor, which means your interaction is specific, okay? And instead of using this wild type version of the competitor, you introduce punctual mutation, and the mutation in which the fragment won't behave as a competitor, it means that you have mutated important nucleotide for the complex formation. But we can come back on that later on. So what I propose you is now we can start to look at this so-called exercise And we're going to see some kind of EMSA like that. So we're going to do first a promoter analysis to find what are the regions of the promoter which are important for the expression. And then we will use EMSA experiment in order to identify the protein DNA interaction. And you will see it's a little bit more complex than this one, than this example. Okay, so I propose you, do you have any question, or we shift to the exercise? We can shift, it's okay. Right, so I change of, so what we are going to, okay, one sec. One second, I lost my. Okay. Okay, do you see the exercise? Okay, so perfect. So we're going to be quite fast for the beginning, because so we are going to study, it's on mammals. We're going to study, it's a quite complete study of the promoter of the growth hormone in mammals. So first point, the growth hormone, it's a very small peptide, so it's directly the product of a gene. So we're going to study the promoter. So this is what is written here. So the growth hormone is a small protein expressed specifically. So this promoter will have a tissue specificity very precise. It is specifically expressed in the anterior part of the pituitary gland. Do you know what is pituitary? It's complicated to pronounce.
Yes,
do you know?
A gland in the brain?
In the brain. It's a very small gland at the back of the brain that is producing plenty of hormones and growth hormone. So we're going to study why this gene, what are the transcription factors necessary for the expression of this GH gene in the anterior part of the pituitary. Okay? So we have two parts in this exercise. The first part is we're going to identify the regulatory elements and the interaction on GH promoter. And then we're going to study the second part are the transcription factors that are governing the expression of the GH gene. So the first part, there is a first question. So first data, they show you in this exercise the sequence of the GH promoter region. So we are not going to answer to all the questions, except if you really want and if you don't know. So first question, it was that was asked with this figure is from a promoter sequence. Can you localize the motifs that are obvious for a promoter? How do you, from what we saw all together, what are the elements, the only elements that you can easily find in a promoter, and not always?
The data box.
That's all. And you see here, there are two evidence, but this is just to have a discussion. At the bottom of the sequence, you've got the numbers, which is the position. So you have the +1, which is here, written in it's 1. You see that there is no 0 position. Everything that is written in minus are promoter sequence. Everything which is positive is coding sequence. And the only thing that we can find is the data box, which is, you see, something between -15 and -20 relative to transcription start. So this was the beginning of the exercise, but it is not what is interesting us. After, are you able? You should be able to answer the second question from what you learn in bachelor. Do you know how to determine the transcription experimentally, the transcription initiation start? Do you know? Or do you have an idea? If you never heard about that, I will come back on that on Wednesday, but we don't have time today. Have you ever heard about the strategy and the methodology to determine the transcription initiation site experimentally? You can tell me, it's not a problem if you don't know.
I'm not sure if I know.
Bon, okay. Anyway, so I will show you on Wednesday this. In order to stay in the promoter study, we're going directly to go, and we will spend a lot of time in this part B. Okay, so we'll see that on Wednesday. So we want to study the promoter of this GH gene. Okay, so look what is written. It's written in order to determine the promoter region. So we call that cis element, the regulatory element, necessary for specific expression in the anterior pituitary. A serial deletion approach of 5' promoter region was undertaken. And so this is exactly what we saw before. So here we've got this drawing, this schematics that show you the different constructs that have been done. So in the previous example, we used the position 220 for the end of the fragment. Here they used position +7. The little black box corresponds to the data box, and you see they cut the promoter of the GH gene little by little from the 5' part, going closer and closer to the data box. Here we are in mammal cells. So the reporter gene that is going to be used in this study, it's not the GUS gene, but it's CAT gene. CAT stands for chloramphenicol acetyltransferase. It's a reporter gene, very frequently used in mammal cells. So they generate this construct. The numbers before the name of this construct correspond to the position, the size of the construct. So for example, PRGH 237 means that the beginning of the promoter is at the position 237 and so on. Okay, so they generate this construct and they clone that in different plasmid, and they use these different plasmid to transfect two types of cells. So this is cell culture, and they monitor the expression of the reporter gene. So first of all, I will explain you. So the left, so what I did for this exercise is I copy and paste the full picture of the paper. But for example, for the reporter activity, in the original paper, they put figure 2 and table 1. Figure 2 for us is not necessary because it's just the way they quantify CAT activity. What is important for us to analyze is the table 1, which is exactly the same strategy as the table we saw before. So listen to me carefully. So they've got different version of the promoter, which is cut little by little, as we saw before. They quantify the CAT activity in two different cell type. They use GC cells, and GC cells, this is cells from pituitary, from rat, that are in culture. So this is cell specific. So this is a cell culture of pituitary cells. Okay, so in this type of cells, the promoter GH should be active because GH is active in pituitary. And the second cell type they use, it's called RAT2. And this is a cell culture from a specific strain of cell, which is coming from liver. So the GH gene is not expressed in liver. Okay, so we are in a situation where we have 4 different constructs containing different version of the promoter, cut little by little, and we test the activity of the reporter gene in 2 different types of cells. One which is pituitary that should express the GH gene, and the other one which is RAT2, so from liver, where we shouldn't find any activity. So all the experiments are summarized in this table. So from that, you are asked to interpret the results. So how would you interpret the results? So I propose you that we can do it all together. So I will share the whiteboard, and it doesn't work. Sorry. 1 sec. Okay. So, what I'm going to do is I'm going to take back this table. So first of all, do you see that whatever the construct used, so 237, 195, and so on, if you look at the expression in RAT2 cells, there is no expression. Anybody see that?
Yes.
Yes.
Okay, so it's the control. None of the construct work in liver type cells. Okay, so we're going to check only for the expression in GC cells. So you see it's expressed in terms of percentage of the control. It's a very common way to present the result. By default, we give the value 100% to the biggest construct. And after we see how this value is going to be modified. So I'm going to write down here, I'm going to draw the promoter. So this is the +1, then we've got the reporter gene, cat. So the first position is 237. So if you have this construct, you have 100% of activity. Okay? Does anybody follow? Okay. If we remove the part of the promoter from 237 to 195, so they make a shorter version of the promoter till the position minus 195. What is happening when you cut the sequence located between 237 and 195? In terms of percentage of activity. And what would be your conclusion?
I see that the expression in GC cells dropped by more than half.
Yeah.
So I think there is something important that lies in that
Okay, so that's
region.
perfect. Yes, you're completely right. We remove a piece of DNA, and what we see that from 100%, we drop, we lose 60% of activity, but does the activity still exist? Yes. Okay.
Yep.
So
Yes.
here, the only thing that you can say is that here there is something that is responsible for high level of expression. So it's more quantitative element. Okay. Do you agree?
Yes.
Okay. And we go on. We have to do lane by lane like that. Then they cut a little bit more of the promoter till the position 141. What's happening? So here we are 100% of activity. If we cut till here, we arrive at 40% of activity. If we remove the sequence from 195 to 141, we drop again. Do you all see that?
Yes.
Okay, so what can we say? We can say here also there is a sequence that is important for strong activity. Okay, this is the only thing that we can say for now. Okay, done. So here we arrive at around 27%. All right. Then we go on and they make the last construct, which is 106. So 106, you see that there is, okay, I put almost 0 because it's the same value, it's 5%. And you see the control without any promoter is 7%, so no more activity. So what would be your conclusion for the sequence contained between 141 and 106?
I think that the important necessary sequence in the promoter for the expression of the gene lies between the minus 237 and the minus 106.
Yes. So this sequence, we can say it's necessary. Okay. We can say that this one, because if we lose this one, we go from an active promoter, even though it's not very strong, to not active, no activity at all. And the two fragments before upstream, they should be responsible for a strong activity. Okay, so from this figure, this is the only result that we can comment, and the only conclusion that we can make. So you will see all the exercise, they are like that. You don't have necessarily big conclusion to draw. So is it okay for you now? And we're going to study more in depth this region in the part number C of the exercise. Is it okay? We go on. So I leave this here because we may need later on. Okay, so we go for the part. So I share back the exercise. Okay. So next part, we're going to study interaction between the trans element, which are the protein factor, and the different region of the promoter. And we are going to use as a method this EMSA that we saw just before. So they explain you that different fragments corresponding to different promoter region of the GH gene were generated by PCR using specific primer, radio-labeled using GAME P32ATP and polynucleotide kinase, and the fragments that have been generated, they are shown in figure 4. So on the top of the figure, in this figure, you've got the promoter sequence. Okay, so here is the transcription start. The fragment used in the construct, they ends at position +7, and they choose different position, not necessarily the same as the one we studied before. So instead of 237, it's 236. Instead of 141, it's 145, and so on. But there are more precise regions as well, and a bigger one. So they generate all these fragments. Okay, and what they show you. So the figure number 4 is only here to show you which region correspond to which fragment. It's okay? And they use all these fragments to do gel retardation experiments, so EMSA, on a gel. And what it has been done using nuclear protein coming from pituitary cells. Very important, because the proteic extract has to be done from the tissue in which the gene is active. Okay, and they show you the result of this EMSA. So, sorry, this one is in French, so it's free probe. Okay, sorry, I change it. And this is complexes DNA of protein on the top. So we have 6 lanes. Okay. In every lane on the top, the numbers on the top are the position, the fragment used as a probe. Okay. So all the fragments are radiolabeled, and they incubate them with extract from GC cells, and they check for the formation of complexes on these different fragments. So what we are going to do is we're going to analyze lane by lane, and I would like you to tell me for every lane what can be the hypothesis in terms of protein interaction. So I need to draw back a bigger region of the promoter. So this is the +1, this is our reference, this is the n +7. So first of all, they used the fragment, okay, sorry, because I was drawing and I wasn't sharing my... Okay, so you see the whiteboard? Yeah, okay. So we're going to look. So first lane on the left, they use the -48. So this fragment from 48 to 7. Do you see any complex formation using this fragment as a probe?
No.
So we can conclude that this fragment, there is no protein interaction, protein DNA interaction. Okay?
Yep. Yes.
Okay. And we go on little by little. So I will keep all the drawing, I will keep like that, so we can make the synthesis of the result. Second lane from the left, they use the fragment which is from 104 to +7. Do you see a complex that can be formed in this when we use this fragment?
I see one.
So we have one spot, okay? One spot. So how would you integrate all these results? The complex is not formed between 48 and +7, but it is formed from 104 to +7. So a quick way to answer would be, we have something that is interacting there. Do you agree? Because it does not interact with the 48 fragment, but it does with 104. Is
Yes.
it? Okay. And we go on little by little. So the third lane, we jump, we will analyze it at the last. We go now for the fourth lane. So the fourth lane, it's the one that is, okay, so I put the position. So there is 1, 4, 5. And 236. Okay. So in the fourth lane, they use as a probe this region. Okay, fragment minus 236 till minus 145. What is there a complex form in this region?
Yes, there's one.
There is one. So we can down like that. There is something that can interact with this region. Okay. Then we go on. We take the fragment a little bit upstream, minus 312 till minus 236. What do you see? Nothing. Do you agree?
Yes.
Yes.
So no interaction. I write down because so we can have the whole map of the promoter. And if we go on for the lane which is completely at the right, it goes till minus 530, and we don't have any complex, okay, formed. So this fragment, nothing, and this fragment, nothing. Okay. Do you agree?
Yes.
Yes.
Okay, now let's talk about the fragment which is the third lane. So fragment minus 145 till plus 7. Okay, so minus 145. What do you see? So look at very carefully, because this one, it's very, very tricky. You see the mouse in the screen? Yes?
Yes.
Yes.
Okay, so this fragment, minus 145 till plus 7, you see in the exercise that you have two complexes that are formed. Do you see them?
Yes.
Yes.
Two different bands.
So two different bands, one a little bit smaller than the other one, but there are two. Okay. So look a little bit, and I would like you to try to analyze this result. So we make the summary. This fragment, the small one, 48, no complex. A bigger fragment that contains the 48 fragment, you have complex formation, and we draw the conclusion saying there is something which is happening between 104 and 48. Okay? Then we use the minus 145 fragment that contains, okay, the 104 fragment, and now instead of seeing one complex, we see two complexes. What can be the hypothesis? Take care, is very, very subtle. But go, we are here to talk. So what I can do in order to understand, I can redraw the fragment, the protein complexes in their respective fragment. Okay, and the green one would be here. All right. So it makes sense that using this fragment, the 104, we have one complex. Why and how can you explain that using the minus 145, you don't have one complex, but two complexes.
I think it's because from the minus 145 and 104, there is another complex.
All right.
I think.
But that.
Yes, maybe.
But what would you say if I said to you, this is not right?
That is also possible.
Sorry, it's not wrong. It's not totally right. Okay, we move a little. I come back on this. Okay, let's take back the example. So this is a gel shift experiment, MSA. And I have a fragment of DNA like that is able to interact with one protein. Okay, basic situation. So in that lane, I don't put protein, and in that lane, I put protein. So if I do an MSA, the probe will move like that. And if I add the protein, I have one complex which is formed. Do you agree with this?
Yes.
Very simple. And this complex here is it is this. Okay? You agree or not? But if this fragment can interact with two proteins, what will we have here? So you have the fragment is this one, two proteins interact with the fragment. How many complexes are we going to see here?
I think if we have two proteins, we still have some like because it's by with the same sequence, it's still one spot on the
exactly.
On
We will have only one spot, and this spot corresponds to the complex of the two proteins. Okay.
So is this the like the protein that I know. I think this was a protein.
So do you have an explanation of what can be the situation that we have where we have two complexes?
So we two different possible interactions.
Yes, but how? It means that we are in a situation with the same fragment, okay, can give rise, can develop two different types of complex. So imagine this. If we have the same fragment here that is able to develop two different complexes with different size because there are different migration, it means that this piece of DNA can develop two different types of interaction. For example, this is an hypothesis. We don't know if it's true, but it means that the lower complexes, the one at the bottom, is the one that has the molecular weight, the smaller, so should be in interaction with only one partner. But the other complexes which has higher molecular weight can be the same complex at the bottom plus a new partner. Do you understand that?
Yes, I also
So if
taught.
we go back in the context of our promoter, so I remove all this. Look again what we found. We found that the fragment minus 104 till plus 7 can form a complex. This is the red protein that we wrote down here. But if we add sequences here, so to go to minus 145, we have in the gel shift in the exercise, we've got two complexes. So it means that this part of the DNA of the promoter, minus 145, can be engaged in two different types of complex. And one of the most possible hypotheses would be, okay, we showed before that the red protein can interact there. So we can imagine that between 145 and 104, there is a second site, but that is not always occupied by the protein. So this would mean that the profile that we see in the exercise, so the first complex and the second complex that we see in the exercise, it can be exactly what we saw before. So this one would be only with red, DNA plus red, and this one would be, we don't know, it's an hypothesis, the red and the blue linked together on the same fragment. So the little difference in the shift will be due to the addition of a second complex to the first one. Is it okay?
Yes.
But what is the other conclusion? I'm sorry, but it's not sorry, but it's one of the most tricky part in the exercise. So if this fragment always interact with the blue and the red, we shouldn't have two different shifts. We should have only the top one, which means always red and blue. But because we have two different shifts, it means that this fragment can be found with two different types of interaction. So it means that the fragment 100, so this fragment minus 145 till plus 7 can exist at the same time in the same cell with two different contexts. The first one would be to have the red partners, and the second one would be to have the blue. So it means that this situation exists alone, and this situation exists also alone. Okay. In other words, it means that this fragment minus 145 can develop an interaction with only the red partner or with the red plus another one. You understand the situation?
Yes.
Okay. So we go on. One second. Okay, one second. Okay, so we go on with the. I just erase this and we're going to look at the next page. We're going to look at figure 6. And what we are going to use, so I stop sharing this and I go back sharing the exercise, and I will explain what is done and we are going to try to analyze these three important parts. Okay, so what do? Here they are still using electromobility shift essay, but now we are using competitors. We are going to use only specific competitors. We are not going to use non-specific, only specific competitors. So you see that there are three gels. We are going to start with one of the two on the right, and then we are going to finish with the one presented on the left because it's the most complicated to analyze. And you see, this is the part of the gel in which we are talking about the formation of these two complexes in this fragment. Okay, so I propose you that we start with the gel in the middle. So the code for reading this figure, at the bottom, this is the piece of DNA which is used for the probe. So this EMS experiment used the fragment -104 as a probe, and on the top, the numbers on the top are the fragments that are used as a competitor. Okay, so we're going to analyze this gel first. Okay, do you agree? Is it fine for you?
Yes.
Yes.
Okay, so I share back my whiteboard, let's say. Okay, so if you use the fragment 104, so I would say it's starting somewhere there. Okay, no, so okay, no, we're not going to see. Oh, I need to put. One second, because maybe I hope I would like to align. Okay. Okay, to align the situation. Okay, so they use this fragment, so -04 till +7 as a probe. Okay, the first lane on the left, you don't use any competitors, and you see that there is the formation of a complex, right? Tell me, because otherwise it's going to be difficult to explain.
Yes.
So, and this is the red complex that we saw before. No problem. If we use, so this is the lane in the middle, the same fragment, 104, as a competitor. Do you see that the complex is disappearing?
Yes.
So it means we are doing a specific competition, because we are using the same fragment as the probe as a competitor. Okay. No particular data there. Then, if we use the fragment -236 till -145 as a competitor, do you see that the complex is still present? Okay, what does it mean? So we need to be very, very precise in the analysis. Okay, so I will try to show you here. So it means that we use, we use this fragment as the probe. We know that there is a protein that is able to bind here. If we use the same fragment as a competitor, the complex formation disappears. Okay, perfect. But if we use the fragment from 236 till -145, so this 236 minus 145, this one as a competitor, you see that the complex is maintained. What is your conclusion on that? I remember you one thing. In the minus 104 plus 7, there is the red protein. But in the minus 236 minus 145, there is the green protein. We showed that before. So if you use the fragment for the competitor, if you use the same fragment as the probe, the complex disappears. Normal. But if we use this fragment that we know that it is able to interact with the green, the complex red is maintained. What can be the conclusion? You may think it's not important, but it's a very, very important conclusion.
I think that I think that the red protein, that means the red protein does not interact with the binding site of the green protein.
The red protein doesn't interact with the green binding site. That's it? You're right, but can you say it in a better way? Okay, so we know that the green protein can bind between 236 and 145. We know that the red one binds to between 104 and between minus 48. Okay, we know that because we are using the two fragments in as competitors, and you see that only the fragment that correspond to the probe can have the complex disappear and not the other one. It means the conclusion is very important. It means that the green protein and the site which is recognized is totally different by the red protein and the site recognized by the red protein. So technically it means red protein is different from green, and red protein recognized a sequence that is different from the sequence recognized by the green.
Yes.
Do you understand? If it was the same protein, the red one here, it would have meant that the sequence here that is recognized by the red protein will be present in this fragment. So if we had used this fragment as a competitor, the complex would have disappeared as well, because if the two proteins are the same, the sequence would be the same, so there will be cross competition. But using the minus 4 as a probe, you have a complex that is deleted with the same fragment as a competitor, but not with this fragment. So, conclusion, the interaction that we see between position minus 104 and minus 48 exists and is totally different from the interaction that we see between minus 236 and minus 145. Do you agree with this? Okay. So for now, we have a little bit more information. We know that green is totally different than the red one. And their binding site as well. Okay, so we know that. Okay. Now let's have a look to the EMSA that is on the right of the page. So the one that is using as a probe the fragment minus 236 till minus 145. So it means we use this fragment as a probe, Actually, what we are going to see, we are going to make the reverse experiment. We use the fragment that is able to interact with the green protein, and we're going to make the competition with the fragment that is able to interact with the red protein. If you look at the result, do they make sense compared to the result we just obtained?
Yes.
So you see on the gel shift, which is on the right of the page, when you use this fragment as a probe, you have, and you add protein, you have the complex which is formed. Okay, we knew that. If we use the same fragment as a complex disappear, okay, makes sense. And if we use the fragment minus 145, okay, so they used this fragment, okay, minus 145. So the one that can be bound to blue and red as a competitor, the complex between the probe and the green is still present. So we confirm that the green protein is not the same as the red one, and we can also say that the blue is different from the green, if the blue exists. Is it okay?
Yes.
So do you see how little by little we are making the model for this promoter and all the interaction with the protein? Okay?
Yes.
So now, anyway, we're going to finish the gel shift experiment, and the second part, the footprint analysis for today. So now we are going to look at what's happening in the gel shift that is presented on the left. So it's the one, so I will try to write it down. So it's the gel shift in which we use the fragment minus 145 till plus 7, so minus 145 till plus 7. So remember, it is the one where we have the blue and the red that is interacting, okay? So we know the position in the middle, it's 104. And we know that for now we have what we call the red and what we call the blue here. And remember that we have an hypothesis in which we can find only the red alone or the red plus the blue together. This is why we have two different complexes. So, first of all, they use this fragment as the probe, and you see that there are two complexes. So, either red alone or red plus blue. If you use the fragment which is the same as the probe, so minus 145 till plus 7, so exactly the same as a competitor, you don't have any more complexes. Does it make sense?
Yes.
Okay. But look what's happening. We are using now half of the probe, so minus 104 till plus 7, and as a competitor. And the consequence, look at the gel, no more complexes. What can be your conclusion? When we use the full length fragment as a competitor, it makes sense completely to have the disappearance of the two complexes, because this is the probe and the complexes, they are going to bind to the competitor. So this is not a problem. But look what's happening if we only block, by using this competitor, we block the binding of the red to the probe. And the consequence is that we don't see any more complexes. So how would you interpret this type of analysis?
I think maybe these two proteins are the same. So when we
Can
have
you speak a little bit louder?
these two proteins are the same. So when we give them only half of the probe, it still can compete with the and
You would say that
protein.
blue and red are the same.
Yes, maybe.
But if blue and yes, okay, why not? Blue and red can be the same, but it doesn't really answer to the question because look what we have before. We know that, we know that when we use the fragment 104, we have one complex which is formed. And remember, when we use the minus 145, we've got two complexes. And we said that we can have only the red on the promoter or the red plus the blue. So I don't think, yes, Telzian?
Yes, I think I'm guessing that maybe the blue protein needs the binding of the red to be able to bind to the probe.
Exactly. Is that because in this context, when you use the minus 104 fragment, you inhibit the possibility of the red to bind, okay? But the consequence of inhibiting, of not allowing the binding of the red does not allow the binding of the blue. So the hypothesis, the best hypothesis at that point is that you have to have the red bound to DNA. Red can bind alone, but you need to have the red bound to DNA in order to have the blue that can bind DNA as well. Does it make sense for you? So we would be in a model which is like this. This region of the promoter, I make a little bit of space. Okay. This region of the promoter, so minus 145, P plus 7. You can be in the middle 104, okay? We can be in a situation where we've got the red in interaction, okay? This is okay. And we can have also a situation where the red is there, and in that case, you can have a blue. Okay, so this is okay, but we can never be in that situation alone. Impossible. Okay, so the concept would be that, and so this is the theoretical model, and this is why we have in the EMSA experiment, we have the free probe at the bottom and we have the two complexes on the top. So this complex with the blue and the red correspond to the biggest complex, and the complex that is formed only with the red correspond to the smaller one. Okay? Does it make sense?
Yes.
Okay. So you see, this kind of electromobility shift assay, you see how powerful can it be, because you can really understand all the mechanistic about the interaction like that. Okay. Do you have any question for this part? So basically, we can say now, for now, we can have so we can remove all this. Huh? This is not important at all anymore. So what we have is what we can do also is to, you remember what is written in blue on the top is, this is the result of the reporter gene activity measures. And this is the result that we have for the gel shift assay. But you will see that we can make a parallel between the two, because this position 106 correspond to almost 104. This one very similar to here. Then what do we have? We have 237, very similar to this. So you see, all this part of the promoter, we said, using the the reporter gene analysis, we said that this region can be subdivided in two, and this is to give strong activity to the promoter. So we can say that the sequence between 236 and something like 150, this is sequence in order to have a strong activity, and this we call that enhancers. So it's to have a strong activity, and all the other part of the promoter, the one that is closer to the data box, with the red and the blue protein, so starting from 150 base pair, something like that, we have this very complex interaction in which we can have only the red one. And if the red one is in interaction with the promoter, then we can have the blue one that can bind. It's a little bit like the red one have a very strong affinity to DNA, whereas blue one has low affinity to DNA and needs to interact together before in order to stabilize the complex. Okay, so this is what we can say about this part of the exercise. All right. Do you have any question concerning this part? Tell me. Anyway, we have 20 minutes left, and we're going to do the second part of the analysis, so we can make the full synthesis on what's happening on the promoter. And this is the footprint, DNA's footprint experiment. So do you have any question concerning EMSA, or shall we go on?
I'm good. I understood.
It's okay for everybody? Okay. So I'm going to share another slide that is coming from one of the slides that you received from Hang this morning. Okay. Just to explain you the methodology of footprinting, which is very simple. Okay, it is this slide. It's one of the slides that I sent you this morning. So what we are going to do, it's called the footprint. There are different methods to do footprint, and the most used is using this DNase. So DNase is an enzyme that cuts randomly double-stranded DNA. And the idea is to have a kind of fingerprint of the protein DNA interaction. So the idea is very simple. The concept is very simple, and you will see that the results are extremely interesting. So the idea is you have a piece of promoter and you know exactly, we are in this situation, we know that there are proteins that can bind to this promoter because we did EMSA experiments before. All these fragments, we label them on one side. It's very, very important. Only one labeling on one side. The labeling can be either radioactivity or other cold probe labeling system like biotin or whatever. Well, anyway, we label one side of the DNA and the same side. So after we do two experiments in parallel, the first one is we don't put any protein, okay? And the second one, we put protein coming from nucleus in which we know the gene is active, okay? So one without protein, the other one with protein. And then, so what is going to happen is that, we can look at this part of the other figure. So the gray part is the DNA. The red balls, they are supposed to be protein interacting with the DNA. And it's called in vitro footprint because we are going to use the capacity of DNAs to randomly cut the DNA, but the DNAs can cut the DNA only in place of DNA where it's not protected by the protein that is in interaction with the DNA. So imagine that all the little arrows correspond to a cut site by DNAs. In the case of the fragment in which we didn't put the protein, the DNAs can cut randomly between every nucleotide. So we have a kind of ladder. And this is represented in this part of the drawing with, if the cut is at the extremity, the fragment is long because we only detect the one that contain the labeling, the radioactive labeling. And you have like a ladder with one base less and less at every ladder step, okay? But if we do the same experiment, but before we put the protein extract, and if in this protein extract there are proteins that are able to bind DNA, they will protect the DNA from the DNase 1. So we will have cuts only in the place where there is no protein DNA interaction. So there is a gap here, and this gap, this is what we call the footprint. The footprint, and it is the place where the transcription factor was bound, and because it was bound to DNA, the DNAs couldn't cut at that place. You see the concept conceptually? So you protect the protein DNA interaction with your transcription factor, protect the DNA from being cut by the DNAs. So you can have a very nice picture on what are the sequences that are protected by the interaction of the transcription factor. Does it make sense for you? Okay. So we're going to use this method. So I stop sharing this and I go back to the exercise. We're going to use this method to understand. So we draw conclusion, very, very strong conclusion using EMSA, and we are going to try to confirm this hypothesis using the footprint approach. So here what we have is the real type of picture of it's not a schematic, it's a real picture of DNA's footprint. So how to analyze this? Hold on, because I would like to have okay. So let's start from the top. From the top, we have three versions of the promoter: the wild type, one mutated version, I will explain you why, and another type of mutation, I explain you why also. On the right here, the number, they correspond to the position of the promoter. So if you, in the promoter, so if you look carefully, here, just in the middle where there is my mouse, it corresponds to the position -100. So technically, the blue protein will bind underneath this, so before position 100, and no, the red one, sorry, and the blue would bind between 147 and 104. Okay, so the top would be the blue protein binding site, and the bottom would be the red protein binding site. Can you see it? So the position on the promoter on the right, and then the numbers on the top. So what I show you are real experiments. So there are some technical consideration to understand. The numbers on the top is the quantity in milligrams of protein that have been added to each lane. Because when you do such experiment, it's so complicated to set up, is that you set up with different amount of protein to find the best one. Okay, so technically it means that where there is 0, it means no protein. So you have 3 cases without any protein, and all the other. So this is from right to left, increasing amount of protein. Okay, it's not a problem. We're going to see. So if it's okay for now, I'll explain you what are the 2 mutations. So HGH wild type is the standard wild type promoter fragment that we used from the beginning of the exercise. LS is a mutation, and it's written here. So it's the wild type fragment, but position from 121 to 126 has been mutated. And the position mutated, if you look at the numbers, 121, 126, it's somewhere there. Okay, so correspond to this gray bar. Okay, and the second type of mutation, it's mutated between -86 and -82. And this is what corresponds to this part of the sequence. Okay, so in other words, the top part, so let's imagine that the separation is around position -100. So the top part is the site of interaction of the blue protein that has been mutated in this mutant, LS121, 126. 120 and 126 is the place where the blue protein should bind. And the 82, 86 is the site, so this one is the site where the red protein should bind. All right? Is it okay? From this experiment, I will leave you 5 minutes. I would like you to tell me if the result presented in this figure are totally consistent with the model that we managed to create using EMSA. Okay, so I leave you a few minutes. Try to analyze this, and tell me what you think about that. So, do you have any ideas? Can you hear me? Yes. Is it okay? Can you analyze this? So, do you have an idea?
I think it's just I can deduce something just simple.
You don't manage to see the correlation with the pre-experiment?
Yes, I think that in the first mutant line where the mutation lies.
But first, before the mutant, for the wild type, what can you see?
Okay, so for the wild type, I can see that the position where the blue.
So the blue is on the top, okay?
Yes, and that gap corresponds to the binding site of the blue protein. So,
okay, perfect.
And yes, I can see that because the blue protein binds successfully to that position, that part of the DNA is not cut by the DNase.
Exactly. Yes.
And the same thing goes for about 90, to 67.
Yes.
That gap is protected by the red protein, so it's not cut.
Okay. So the result, if we make the summary, the result that we have in the wild type, as you said, it's perfect what you said. We found back the idea that we have the red protein that can bind to what was predicted, and the blue can bind also. Okay? Yes or no? Is it okay?
Yes.
Okay. Now look what we do here. First type of mutant. We are in the 121, 126, we are mutating the site of the blue binding, the blue protein binding site. So we have to choose one concentration of protein that is the same, or otherwise it's complicated to analyze. So let's take, for example, the lane, we are going to compare only the lane labeled 12. So it means 12 microgram of protein, so we can make comparisons. So look, if we are in the wild types, so wild type promoters, when we use 12 microgram of protein, the blue can bind and the red can bind. You agree?
Yes.
If we mutate the site of binding of the blue protein and use 12 microgram of protein, the blue protein cannot bind. You see, it's not protected anymore. You see, but the red can bind. Do you agree?
Yes.
And if we do the same where we mutate the red protein binding site, okay, so you see with 12, or 12, actually it's not the right, let's take 6. Okay, I start again with 6. This is why we put different amount of protein. With 6 microgram of protein, when you mutate the blue binding site, the red protein can still bind. But when you mutate the red binding site, okay, the red protein does not bind anymore, which makes sense, but the blue one does not bind anymore as well. Can you see it?
Yes.
So, does it confirm the model?
I think so, yes.
I share back the whiteboard. Okay, so we had this model from the beginning. So this is the red and the blue, and the consequence of so I remove this to have a little bit of space. So what we use for the footprint, the result, they are a little bit this. If we do not mutate none of the sides, there is a protection for the red side and for the blue side. Okay, if we do a mutation only in the site of the blue binding protein, we still have protection for the red site. But if we mutate the binding site of the red protein, you don't have anymore the blue binding site, the blue protein binding site capacity. So it means that, is it okay for this conclusion? So it is completely supporting this hypothesis in which the red can bind alone, but to have the binding of the blue, you need to have the red bound first. And this is why in the electromobility shift assay, we have the two complexes that correspond for the lower one with interaction with red only, and for the top one, it's red plus blue. Okay? Is it okay? So we finished this part of the exercise. So do you have any question? And do you see that it's using very simple method that we can have very detailed information even on the mechanistic of interaction, because in that example, which is very nice, you see that there is a binding which is necessary to be established first in order to have the second interaction that can be developed. And it's the way this promoter is working. Is it fine? It's
Yes.
okay. Okay, perfect. So it's 12. It's midday for me, so it's 6 p.m. for you. So we need to stop now. Do you understand everything in this exercise and the parallel with the lecture that I showed you before for the CCR gene and the promoter interactions? Is it fine?
Yes.
Okay, so what we are going to do, so tomorrow we don't have lecture altogether because I have another lecture in France, which is complex. We will see back all together on Wednesday. And we are going, so what we are going to do is I'm going to explain you how from a promoter analysis like we did today, we can isolate how we will do the molecular characterization of transcription factor. And we will come back to the end of this exercise, which is the case study for studying the transcription factor that are going, so in other words, we are going to study the transcription factor that bind to the red protein, the one which is bound there. And so on Wednesday, I will explain you how from this type of data, so promoter data, we can clone the cDNA corresponding to the transcription factor. And we will use this exercise and another example to show you how we can work with transcription factor and to know how they can control transcriptional activity. Okay,